Hello students, have you ever wondered how a tiny seed can grow into a towering tree or a vibrant flower? Today, we are going to explore the fascinating world of seeds, the very foundation of plant life. A seed is the reproductive unit of a plant. It contains a tiny, developing embryo, a protective seed coat, and stored food reserves to nourish the young plant during its early stages. Think about the seeds you plant in your garden. Those little grains eventually grow into fully developed plants like beans, tomatoes, or sunflowers. Seeds are vital because they ensure the continuation of plant species and allow plants to spread over long distances. The seed coat, the outer layer, acts like a shield, protecting the embryo from physical damage and preventing it from drying out, just like a baby relies on the safety of a mother's womb. So next time you hold a seed, remember, it's a miniature package of life, ready to unfold its potential under the right conditions. Let's take a closer look at a dicot seed, like that of a pea or gram. The seed coat forms the outermost layer and has two parts, the testa, which is the tough outer layer, and the tegmen, the delicate inner layer. On the seed coat, you'll notice a scar called the hilum. This is where the seed was attached to the fruit. Nearby is the micropyle, a tiny pore through which water enters during germination. Inside the seed lies the embryo, the young plant waiting to grow. It includes the radicle, which will become the root, the embryonal axis, and two cotyledons, or seed leaves. In dicots, these cotyledons store food, providing the energy the embryo needs until it can make its own food through photosynthesis. So, every part of a dicot seed is perfectly designed to protect and nourish the next generation of plants. Now, let's examine a monocot seed, such as corn or maize. Here, the seed coat is thinner and often fused with the fruit wall. Inside, the endosperm is large and stores food for the developing embryo. Separating the embryo from the endosperm is a thin layer called the aleurone layer. The embryo of a monocot is different from a dicot. It has a single, large cotyledon called the scutellum, a short axis, a plumule that will become the shoot, and a radicle that becomes the root. In corn, the large endosperm provides all the energy the seedling needs to grow. Isn't it amazing that such tiny structures are so carefully organized for survival? Have you ever seen a bean sprout in a damp paper towel? That's germination in action, the process by which a seed develops into a new plant. For germination to start, a seed needs water, oxygen, and the right temperature. The first structure to emerge is the radicle, which grows downward to form the root system. The cotyledons provide stored food to the young seedling until it can make its own food through photosynthesis. Watching germination is like observing a miracle. From a tiny dormant seed, a living plant begins to emerge, fueled by nutrients packed inside it for this very moment. Let's recap what we've learned about seeds. From fertilization to seed formation, the seed is the next stage in the life cycle of a plant. Understanding the differences between dicot and monocot seeds helps us appreciate the diversity of plant life on Earth. Seeds also play a crucial role in ecosystems. They sustain plant populations, provide food for humans and animals, and contribute to ecological balance. Remember the saying, "From small acorns grow mighty oak trees." Seeds hold the incredible potential to create life, forming the foundation of the plant kingdom. So, next time you see a seed, think about the intricate processes it carries within: protection, nourishment, and the potential to grow into a new living organism. Biology truly is the science of life in all its beautiful complexity. I hope this session opened your eyes to the amazing world of seeds. See you next time as we explore more secrets of plant growth and development.